So, concerning the CAO seminar tomorrow, Aviv Solodach will be giving the lecture at 11 o'clock. It will be called Surprises in Ocean Ventilation through the Antarctic Margins and Density Layers. At 10 o'clock, there will be a PhD seminar, PhD lecture being given by Shalom. That will be called The Hydrological Processes in the Nevada Zone under anthropogenic pressure. I think that's in room 126, but I'm not 100% sure. Number 3 is on Sunday, the 9th of the 11th at 10:30 at the Geological Survey. Yuval Bartov will be talking about subsurface mapping in Israel and implications for hydrocarbon exploration. Today, after this seminar, there won't be the usual meeting that we have. We'll get together at 12 o'clock. But we will be going for that next week. And now I'd like to introduce our speaker for today, Shlomit Sharoni. She did her bachelor's at Hebrew University. She did a master's and PhD at Weizmann. PhD was with Itay Halevi. And she's been doing her at MIT. She's been doing her postdoc for the last 3 years. And we'll be hearing about her research from that period today.
Thank you, Simon, for organizing the conference. Sorry, I'm a bit excited. So, my name is Shlomit, and today I'm going to talk about modeling the macromolecular and elemental composition of the ocean. I will start with a few words about myself. I'm a biogeochemist, and I develop models to explore the interaction between life and its environment on multiple times. So I did my bachelor in chemistry in the Hebrew University. Following that, I pursued a master in a more marine biology-oriented lab at the Weizmann Institute, where I grew phytoplankton cultures, and I also did some field work. For my PhD, I shifted to a more quantitative PhD at the Weizmann Institute, where I developed biogeochemical models to explore the coevolution of life and its environment on geologic timescales. And I'm currently a Rothschild, Fulbright, and Simon postdoc fellow at MIT, working with Professor Mick Follows, where I develop Earth system models to explore the impact of cellular level processes in marine phytoplankton. So as you can see, I have a diverse background from tools in molecular biology to 3D ocean models. And this equipped me with a unique perspective to tackle questions in biogeochemistry, a field that is by nature multidisciplinary and complex. And the talk outline, I will start with a brief introduction, motivation to my work. I will briefly introduce you some of my PhD work, and I will mostly focus on the work that I'm conducting now at my postdoc at MIT. And I will give you my future perspective. I'll speak about the future perspective. But just a few words about my scientific approach. So I like to ask global scale questions that are related to the role of marine phytoplankton, how they impact global scale phenomena like the ocean carbon sink, ocean deoxygenation, etc. To tackle these questions, I developed biogeochemical models. And I developed these models based on deep exploration of laboratory experiments and field observations. So I developed new parameterization, and I extract parameters that are based on real data. A crucial part of my work is to compare my results with global observations, and I use oceanographic, geologic, and biological observations. It depends on the question I ask. And hopefully I'm discovering new things about nature. So this process is not linear. Sometimes I discover new things by the model development or by the comparison or the mismatch between model and observation. So I will show you how I developed my PhD according to this scheme, and then how I developed my postdoc. I will start with a brief introduction to my topic. So, life and its environment tightly interlinked. Plate tectonic creates environments such as mountains, ocean, mid-ocean ridges, niches where life can evolve and diversify through time. On the other hand, life highly impacts its environment. Perhaps the most notable example is the activity of this unicellular microorganism named phytoplankton. These photosynthetic organisms are responsible presently for 50% of the global annual net primary productivity. By utilizing the solar energy, they convert inorganic elements such as carbon dioxide, nitrate, and phosphate, and they produce oxygen. And in this way, they redistribute these elements between the ocean atmosphere and solid earth reservoirs. One of the greatest manifestations on the way phytoplankton impact their environment is the concept of the Redfield ratio. So Alfred Redfield is a famous oceanographer from Woods Hole Oceanographic Institute, and approximately a century ago, he measured the dissolved nutrients concentration in the deep ocean in many locations around the globe. And he found that the dissolved nutrient concentration, for example, the nitrates, dissolved nitrates to phosphates in the deep ocean, the ratio of them is approximately constant all around the ocean. And when he measured the ratio of these elements in the organic cells of phytoplankton, he found that this ratio is constant to the ratio of the dissolved inorganic nutrients in the deep ocean. So the ratio of the dissolved organic nutrients in phytoplankton cells that live only in the surface of the ocean impacts the ratio of the dissolved nutrients in the deep ocean. And this is because phytoplankton, when they sink, the organic matter gets respired, and in this way they convert the organic into the organic form. So since this is a very key concept in oceanography that any first year student oceanographic study of the constant Redfield ratio. So although Redfield acknowledged that there are variations in this ratio, a constant Redfield ratio of carbon to nitrogen to phosphorus in the organic matter of 100 to 16 to 1 is a frequently used parameter in biogeochemical studies and in biogeochemical models. So this ratio is the ratio of carbon to nitrogen to phosphorus in phytoplankton cells. And you might ask why this ratio is so important. Well, this ratio regulates the ocean's carbon storage. Okay? If phytoplankton cells will increase their carbon to nutrient ratio, more carbon will be sequestered via the biological pump, via the phytoplankton, and more carbon will be stored in the ocean and less will be in the atmosphere. This ratio also regulates oxygen production, how much oxygen gets produced in the surface ocean by phytoplankton, and it also regulates how much oxygen gets consumed in the deep ocean due to respiration. So there are many regions in the ocean where oxygen is too low to sustain aerobic life, and this ratio regulates the sink, how much oxygen gets consumed. Furthermore, phytoplankton form the base of the marine food web. So this ratio regulates how much nutrients and how much energy is stored within the organic matter and how much is available for the entire marine food web. So this ratio is important for biogeochemical studies. So although a century after, many observations have accumulated to show that this ratio is not constant on the Redfield ratio, and that there are global variations in this ratio. These are samples that were collected across different oceanic basins, and these are measurements of carbon to phosphorus in organic matter, and you can see that it's not fixed on the Redfield, like commonly assumed, but there are global variations. It is higher in the subtropical or oligotropic gyres, and lower at equatorial regions and high latitudes. So the red fill ratio is not fixed, it's variable. So there are large-scale variations, seem to be a function of sea surface temperature and phosphate concentrations. However, the causes for this global variability remain unknown. So despite these global variations that we know that now exist, most of the biogeochemical studies still use a fixed stoichiometry, and as I said before, it can change the amount of carbon that is sequestered via the biological pump, how much oxygen gets consumed, and the amount of nutrient and energy that are available for the entire marine ecosystem. So one of my PhD questions was to challenge the assumption of a constant red field, also in geologic time, because also paleoclimate models that simulate past environmental conditions also assume a constant red field ratio. However, we know that environmental conditions in the past were very different temperature, different nutrients availability in the ocean. So I challenged the assumption of a constant red fill ratio, also through geologic time. So this was my global scale question. What was the paleo red fill ratio? To explore this question, I developed a mechanistic model, developed a geologic model. I call it the BioGeo model. This model resolved the cycling of different elements in the ocean atmosphere and in the sedimentary rock. To resolve the cycling of phosphate, oxygen, calcium, and carbon and alkalinity in the ocean atmosphere and in the rock reservoirs. And I wanted to apply the present day correlation between the red fill ratio to environmental conditions, but first I investigated what are the factors that control this ratio in the present ocean, in order to understand that if I can apply it to the past, maybe the present day correlations cannot be applied to the past. So I studied what causes the global scale variation in the red fill ratio, and I found that these are due to changes in phytoplankton community compositions. So there are many phytoplankton groups in the ocean, and they have different elemental composition. For example, diatoms that grows in high latitudes have lower N2P in the organic matter relative to cyanobacteria, for example, that occupy the subtropical gyres. So I found that the changes in the community composition of this phytoplankton regulates the variation in the CTP. And since this group existed in the past, I could apply this parameterization also to the geologic past. I will not show you now, but I compared and validated my model against geologic processes such as PCO2, delta C13 in sedimentary rock, and iron to phosphate ratio. And I extracted many parameters also by conducting extensive literature review. And I discovered something new. So I found that the red fill ratio was likely not constant throughout the geologic past, but it was, okay, so let me explain you this graph. This is age in million years. This is the early Phanerozoic, the eon where life started to appear in the geologic record. And this is the elemental composition, the carbon to phosphorus ratio in organic matter, and it's usually assumed to be constant. And I found that it was likely not constant, and it changed in response to several geologic and evolutionary events. For example, during the Devonian, you had the evolution of land plants, and during the Mesozoic, you had the formation and the decomposition of the supercontinent Pangaea. And these evolutionary and geologic events change the amount of nutrients that are delivered into the ocean and also change the climate. And they drove a reduction to the present day red fill ratio that we see today. So the paleo red fill ratio was likely not constant, it was likely higher, and it declined to the present day red fill ratio due to several geologic and evolutionary events. So during this process of my PhD, I discovered several new things. First, by the development of the model, I found that phytoplankton elemental composition is phytoplankton group composition of well-adapted phytoplankton dictates the global variation in the ocean. I found that the elemental composition was higher than the red fill ratio throughout the geologic past. I didn't discuss this part, but this was another exciting discovery during the exploration of my model. I found the global phosphorus cycle, that is phosphate is an essential nutrient for the marine biosphere, that the global geologic phosphate cycle also sensitive to a process called the temperature dependence of seafloor weather. And I know some of you are working on this, so I'm happy to discuss further if you. Okay, so now I will speak about my postdoc work. Okay, first I want to explain you why I went to MIT and I joined the group of Professor Mick Follows. So Professor Mick Follows and his group members developed the Darwin model. The Darwin model is the most cutting-edge ecological and biogeochemical model that is currently embedded in the general circulation model. So this model can simulate the growth of hundreds of phytoplankton groups that are highlighted here by the different colors. It can simulate different ecological interactions like as phytoplankton, zooplankton, viruses, fish, bacteria. And it can simulate the distribution of many tracers, carbon, oxygen, phosphate, nitrogen, iron, silica. Choose one element that you want and we can develop its evolution in the model. So this model is called the Darwin model because my postdoc advisor showed that this model can simulate an evolutionary process of natural selection. For example, what they did here in the beginning of the simulation, they seed hundreds of phytoplankton groups in very low concentration in each grid cell. Now, when they run the model forward in time, the biogeography of the phytoplankton groups emerged, right? So, and it was in agreement with global observations. For example, the red group here is diatoms. It's known that they occupy the high latitude, and the model was able to simulate them. And so the model captures the biogeography of phytoplankton groups. And I was already, when I finished my master's, I wanted to go to MIT to learn to develop this model and to be able to use it in order to apply my own questions. So when I joined my group, my goal was to continue to develop this model to include variation in the macromolecular composition of phytoplankton. So I know that you have a very diverse background, and I'm going to speak a little bit on biology, so you can stop me and ask if you want. So this is a phytoplankton cell, okay? And different phytoplankton are composed like us, like many living organisms, mainly from proteins, also from carbohydrates, from lipids, from pigments, nucleic acid, RNA and DNA, and other macromolecules. Now, each macromolecule has different elemental composition. For example, proteins are rich in nitrogen, and carbohydrates and lipids don't have any nitrogen. Lipids are also more reduced. They contain a lot of hydrogen and carbohydrates contain more oxygen. So the relative proportion of these macromolecules in the cell can regulate the elemental composition of the phytoplankton, which can regulate, again, how much carbon is sequestered in the deep ocean, how much oxygen gets consumed, and how much energy and nutrients are available for the marine ecosystem. So, and we know the different macromolecules change. It's not constant. They change as a function of environmental conditions. For example, under low light conditions, phytoplankton increase their chlorophyll concentration in order to produce more antennas to capture more efficiently the light. Another example is here, under nitrogen-free conditions, phytoplankton don't have enough nitrogen to produce proteins, so they reduce their proteins level and increase their carbohydrates and lipids at the expense. So we know that phytoplankton macromolecular composition can change in response to varying conditions, and variation in phytoplankton macromolecular composition under varying environmental conditions can alter the amount of energy and nutrients available for the marine ecosystem. So understanding, resolving the macromolecular composition of the global ocean is important to understand how much energy and how much nutrients are available for the entire marine ecosystem. So again, to answer this question, I now developed the Darwin model, the model that I showed you earlier. So the first generation of the Darwin model developed by my postdoc advisor, included a fixed elemental composition on the Redfield ratio. The second generation of the model included a variable carbon to nitrogen to phosphorus. However, it's assumed in this version that these elements are within a separate compartment in the cell, which is not very physiological realistic, because we know that the different elements are within different macromolecules. So the generation that I currently developed include variation in the proteins, lipids, carbohydrates, nucleic acid, and other storage macromolecules as a function of environmental conditions. So my development of the model is currently part of the main branch of the MIT General Circulation Model, MIT GCM, and my specific modification can be found in my GitHub. Some of the assumptions of the macromolecular models. Okay, so the macromolecular model assumes that this is the carbon in the cell, that it increases due to photosynthesis, which is a function of light, hyperbolic function of light. It decreases due to growth rate and respiration. So cell carbon increases due to photosynthesis and decreases due to growth rate, due to respiration and growth rate. We assume that the cell carbon overall on average remains fixed, and we can extract the chlorophyll to carbon ratio as a function of photosynthesis, growth rate, and respiration. Another hypothesis of the assumption of the model is that the composition of the photosynthetic compartment is fixed. So if we know the chlorophyll to carbon ratio, we can calculate how much photosynthetic proteins we have and how much phosphorus, for example, in the thylakoid membrane. Another assumption is that the RNA to protein ratio is fixed. So most of the RNA in phytoplankton cells is in ribosomes. Ribosomes are proteins that are used to grow. So if phytoplankton cells grow faster, they will have more ribosomes, and therefore the RNA to protein ratio increases. The fourth assumption is that the non-limiting nutrient is stored in the cell. My ecological setup. So I have two phytoplankton groups, one small, broadly representing cyanobacteria with high surface area to volume ratio. It can acquire nutrients at low nutrient concentrations. The second phytoplankton is large phytoplankton. It can store a lot of nutrients. It can grow faster. I have a single grazer that grazes upon both, but preferentially like the small ones. So this ecosystem is growing as a function of light, nutrient, and temperature. And also the macromoleculars in the phytoplankton cells change in response to these conditions. My tracers are being transported and mixed within the IGSM. The IGSM is an Earth system model of intermediate complexity. It has a 3D ocean with a 2D zonally average atmosphere. The ocean model is the MIT General Circulation Model. So this was developed by many groups at MIT. So the ocean model has a resolution of 2.5 longitude and 2 latitude. It has 22 vertical layers, 10 meters at the surface, 500 meters at depth. So I simulated my tracers with this physical field from this model, velocity field from this model, and I conducted the pre-industrial simulations and also climate change simulation to understand how this will change in the future. And the climate change simulation are done with a human activity model that simulates the amount of emission as a function of population and economical growth. Now I will share with you some of my results. So these are amount of proteins in phytoplankton cells. It's percentage. It's a fraction, sorry. So 0 it means that cells have no proteins. 1 it means that they have only proteins. So you can see, and this is a depth integrated mean of pre-industrial simulations. I will show you the high climate later. So this is proteins, 0 is blue, 1 is red. So you can see that there are spatial patterns of the amount of proteins in phytoplankton cells. Phytoplankton invests less in proteins in the subtropical or eutrophic gyres. These regions are called the oceanic deserts. In these regions, phytoplankton are exposed to high light, and they have not a lot of nutrients. These regions are very limited in nutrients. So phytoplankton don't have enough of nutrients to produce proteins, so proteins level are lower. At the high latitudes, phytoplankton have more proteins, and this is because phytoplankton in high latitude have less light. They need more proteins to capture more efficiently the light. And also they have more nutrients, so they can grow faster, so they can have more proteins in order to grow faster. And the carbohydrates and lipids have a mirror image of the protein. So in the subtropical gyres, where phytoplankton have low nutrients and high light, phytoplankton dissipates the excess of energy in carbon-rich macromolecules such as carbohydrates and lipids. Now, we all know the different macromolecules have different caloric content, right? So lipids are much more caloric dense relative to proteins and carbs. So by applying an average value of caloric value, I can estimate what is the caloric content, the organic matter at the base of the marine food web. And here you can see a global map of the energetic content at the base of the marine food web, and you can see that in the gyres, the energy content is slightly higher, and this is because you have more lipids in these regions, which are caloric dense. So despite the fact that you have less biomass in these regions, the caloric content is higher in the subtropical gyres, and it's lower in the high latitude. Next, I compare my model to available observations of macromolecular composition. So this, for example, samples were collected by the FRL. They conducted a transect in the North Pacific, and they sampled the macromolecular composition of phytoplankton, and they found that this is latitude and this is percentage. So there is a positive trend of proteins with latitude. Toward the high latitude, phytoplankton invests more in proteins, less in carbs and lipids, and this is broadly captured by our model. Of course, the model is more, we have less noise because the model is coarser resolution, but the trends are broadly captured. There is also a large transect conducted by Handa et al. in 1973 that show a negative trend with sea surface temperature of proteins, and this is also captured by our model. So our results are consistent with sparse observations of macromolecular composition. So there are not many measurements of macromolecular composition, but there is much more measurements of elemental composition. So these are gray dots are many measurements that were collected around the ocean, and not by us. They were collected by Tanioka and Martini. And you can see the latitudinal trend here in gray, and the map showing our model results, which is the latitudinal trend here in solid line. So you can see that there is good agreement between the model and observations. And again, observations are very noisy. They are conducted at different seasons around different locations, but overall our model shows good agreement with the observations. Okay, and now I will show you what is the projected change by the end of the 21st century under the high emission scenario, assuming that the human will continue to emit CO2. This CO2 will reach in the atmosphere approximately 850 ppm. So our model projects first changes in the environmental conditions. This is sea surface temperature versus time. So you can see that by the end of the 21st century, our model projected an increase by approximately 3 degrees in the sea surface temperature. This is an amount of nitrate in the surface ocean. How much nitrate relative to the pre-industrial conditions. And here you can see that the nitrate in the surface ocean decreases by approximately 11%. And this is because you have the extra heat that is captured by the surface, enhances stratification of the water column. And therefore, you have less nutrients that are delivered from the deep ocean. So temperature increase and nutrients into the surface ocean reduces. These results are in agreement with other Earth system models that are part of the CBIP6 models. But our new result is that we project how phytoplankton physiology will change in response to these conditions. Okay, so this is anomaly plot, right? Blue mean less, red mean more, and this is the amount of proteins in phytoplankton cells. Okay, and this is depth integrated cellular allocation to proteins. So our model projects geographical variable responses, right? So first, let's focus on the subtropical gyres, okay? Here we got really unintuitive results. So I showed you before that concentration of nutrients is going to decline because of enhanced stratification. However, our model projects that phytoplankton will have more proteins. I showed you before, if you remember, that to produce protein you need nitrogen. So how come if nitrogen is declining, phytoplankton have more proteins? This was very unintuitive results for us. So therefore, I'm now going to show you a depth profile in this region, in the North Pacific, subtropical Pacific. Okay, so this is the depth in the water column. The heat map is temperature, so it's warmer in the surface and it's cooler in that. Okay, and you have this is the depth of the photic zone where light is 1% of the input. So this is a classical vertical profile of phytoplankton. So you have less nutrients in the surface and more at depth because they are coming from the deep ocean. And phytoplankton biomass are in sweet point between enough light from above and enough nutrients from the deep. So this is a classical profile, and this is in the pre-industrial condition. Okay? And phytoplankton that are in this depth, they're exposed to high light. They don't need a lot of photosynthetic proteins because they have a lot of light. What happened in the end of the 21st century? So we have warming. It will enhance stratification. You have a more mortality of phytoplankton in the surface, so more light penetrates. And the enhanced stratification reduces the amount of nutrients that are delivered from the deep ocean. Therefore, phytoplankton biomass shifts a bit deeper in the water. Also, it's warmer for them there, and they want to be closer to the nutrients. So the biomass shifts deeper. This deeper point, phytoplankton have more photosynthetic proteins because they need more to capture the lower light in this region. So this is an important, interesting phenomenon. I spoke then with Oli about it because it means also that phytoplankton also absorb heat, right? So it can have an interesting biophysical feedback about the depth of the mixture because if phytoplankton biomass shifts deeper, it means that it can change the profile of the temperature in the ocean. So this is a very interesting phenomenon, and it's already been shown in the BATS station in the Sargasso Sea and also in other places that the biomass indeed shifts deeper. And we showed that this causes an additional protein. So again, this is year, and this is biomass. So the surface biomass declines with time because it's hotter and because there is less nutrients. Subsurface population increases with time. Subsurface population of more proteins. Therefore, the overall depth integrated response is more proteins. Okay. Great. So I hope I explained you why phytoplankton in the subtropical gyres have more proteins. Now let's focus on the polar regions. In the polar regions, our model projects that phytoplankton will have less proteins. What's happened in the polar regions? So you have warming. Warming reduces ice cover. Therefore, phytoplankton will have more light because less ice, more light. So because they have more light, they need less photosynthetic proteins to capture the light. So photosynthetic proteins reduces because phytoplankton will have more light because they have less ice. This is the mechanism in the polar regions. It's not only this, there is also other mechanism because the warming also enhances growth, which enhances photosynthetic protein, biosynthetic proteins. However, the overall total response is that phytoplankton in the polar regions will have less proteins. This is the carbohydrates and lipids, and you can see again a mirror image. Okay, so a crucial part of my work is to compare the data with global observations. However, it's not so easy to do it in oceanography because many observations are sparse in time and in place. And for example, so I conducted a database, a new database of macromolecular composition in the ocean in marine phytoplankton. So this database contains 45 studies of in situ macromolecular measurements across the ocean. So you can see that many of the measurements are in restricted basins or coastal environments, locations that global models don't reproduce very well, don't capture well. And 50% of the study were in polar regions, in the Arctic, in the Antarctic, because of the significance of the polar regions for primary productivity, for fisheries. So 50% of the studies are in polar regions. So when I tried to compare my model against observations, I did not look for a temporal trend. I did not think that we are going to see climate change already 2025. So I didn't look for a temporal trend, I looked for a spatial trend. However, the trends in the poles were all over, and I could not find any correlation. And only until I plotted the samples as a function of time, I saw a trend. So what I found in this database, that phytoplankton in the Arctic, so they have been sampled from the 80s already. We have 40 years of data from Arctic regions. So I found the phytoplankton in the Arctic, the proteins in phytoplankton is declining, and the carbs and lipids are increasing with time. Of course, just think about this is biological measurement, environmental measurement, there is a lot of noise, a lot of variability, but still the trend is statistically significant. Phytoplankton in the Arctic regions have already less proteins and more carbohydrates and lipids, and these are measurements, this is not model. In the Antarctic, we find something similar. Phytoplankton have less proteins, more carbs and lipids. Also, and this is in 40 years, we already see this trend. And this makes sense because the Arctic, the polar regions are warming much faster than the rest of the ocean. So it means that we already starting to see the impact of climate change on the physiology of phytoplankton in these regions. And this is in agreement with my model that show the mechanistic why phytoplankton reduce their protein and increase in carbohydrate. So that means that phytoplankton macromolecular composition is not only important for global biogeochemistry and for ecological reasons, but it can also serve as an indicator for changing environmental conditions. Okay, I will briefly go over the implications of my model. So I found that proteins overall in the most productive regions are going to decline. Energy content is going to increase because they will have more lipids and more carbs. Lipids are caloric dense, so energy increase. The nutrients get diluted. The C2P and C2N increases also. So in a way, my model predicts that the food in the base of the marine food webs will get more junk food. It will be more caloric, it has more calories, but it will have less protein and less nutrients. And in a way, this process echoes a process that's already happening in the terrestrial ecosystem, where plants, because of the high PCO2, they dilute their nutrients because the carbon to nutrient ratio increase because they take more CO2 from the atmosphere. So while the mechanism in the terrestrial system is completely different, plants diluting and become more junk food because of higher PCO2, the mechanism is completely different in the ocean and is driven by changes in temperature, light, and nutrients. So how the impact of the marine food web. So some experiments have shown that fish that are fed on Daphnia, that are fed on phytoplankton with low quality, the biomass of the fish is highly reduced. So it means that it has significant implication of the food web, which can also transform to us, to humans that are fed on fish. However, some copepods that live in polar regions may benefit. So this copepod has evolved strategy to extract lipids from its food to survive the prolonged periods of darkness in the polar regions. So this copepod may benefit from higher caloric energy in the poles. But these are still open questions that I left to explore. So I found that under projected warmer climate, phytoplankton in subtropical latitudes are projected to increase their protein content because of subpopulation of phytoplankton decrease. At polar regions, cellular protein levels are projected to decline. I already see it in observations of 40 years. It's already happened. And such changes will likely reshape the nutrition landscape at the base of the marine food web and other global biogeochemical cycles. So this paper is currently under advanced review, second round of revision in Nature Climate Change. I hope to soon get accepted. Okay, I don't have so much time. I'll just briefly go over another project that I'm doing that's related to ocean deoxygenation. So unlike the atmosphere that is fully oxygenated, there are many regions in the ocean where oxygen is too low to sustain aerobic life. These regions are here, highlighted in red. So oxygen gets delivered to the deep ocean due to physical transport, and it gets consumed due to respiration of organic matter in the ocean. So many models predict that the oxygen concentration in the ocean will reduce in the future. It's already observed because of reducing physical mixing and because of warming that reduces solubility. However, less attention was given to the role of changes in the phytoplankton. They can also change in response to climate change, and how these changes will either mute or propagate changes in oxygen in the oxygenation. So this is what the second project that are currently in bioarchive. So I asked what is the sensitivity of the global oxygen cycle to changes in the biochemical level, in the oxidation state of the organic matter. So lipids are much more reduced, contain more C2H bond relative to carbohydrates. So I asked what will happen to the oxygen cycle if the cell will shift from carbs to lipids. So more oxygen will get consumed when when lipids are expired. So I found that the extent of the OMZ are sensitive to variations in the chemical properties of the organic matter. For example, if you increase the cell will increase its protein level, the OMZ will expand because more oxygen will get consumed. And to understand this question about the oxygen cycle, I developed the Darwin model to do a long simulation of 2,000 years. So most of my group at MIT focused on the surface ocean, the 100 meter, and because they are more interested about diversity, about ecological questions, and because I'm interested about understanding fluxes such as POs, organic carbon flux, DOC, oxygen, I developed a platform to simulate the model for 2,000 years. That is the time that it takes for the oxygen to equilibrate in the deep ocean. And you can see here a vertical transect of the Southern Ocean, and model the solid line, and these are the observations. So I am able to simulate well the vertical profiles in the ocean. So I'm going to continue and use this platform to ask in the future. Okay, so now I'm going to explain what I'm going to do in the future. I will continue to ask what are the biogeochemical consequences of macromolecular variation on the future ocean. What is the consequences for carbon export, for ocean deoxygenation? How do the macromolecular transform as they move through the marine food web? So we know that what ultimately gets preserved in the sediment is mostly lipids. It means that there is preferential remineralization or respiration of macromolecules through the food web. And this is another goal to try to understand this transformation. Also, viruses and grazers impact the stoichiometry of marine organic matter because they have different elemental requirements. I'm going to include these interactions in my model. And I also want to focus about the biological factors in ocean deoxygenation, which is an area that is kind of neglected. So I will continue use models that I've developed, the geological model, the Darwin model, and also other, but I want to develop new biogeochemical models, possibly with the collaboration with this department regarding the Moses Observatory project, and also to quantify marine carbon dioxide removal approach in collaboration with department members from here. We also need to collect more data, as I showed you. There is not a lot of data enough data to validate and prepare my model. I want to work with collaboration to collect more data and to analyze it. And to discover new things about nature. And with that, I want to thank, this is is not part of my group. This is a meeting in New York. This is my postdoc advisor, Professor Nick Follows. And this is me. I want to really thank him. He's really kind and smart and very positive person. And I'm really grateful for having me as a postdoc. I want to thank my funding fellowship, Fulbright, Rothschild, and Simons. And these are my manuscripts. You can find them in the archive and on our website. And I want to thank you for listening, and I'm happy to take questions.
Questions?
I wanted to ask about internal variability. So you showed these observations of a trend in the high latitudes. So let's say that you take, you say enforced only by natural forces. So when the long-term pre-industrial run, what variability would you get with just internal? I mean, in order to attribute this change, this trend to climate change, you need to compare it to some natural variability and show that it's outside of the natural range, right?
Yeah.
How large, do you have a feeling of how large?
The internal variability?
Yeah. Or changes due to internal variability in the ocean?
You mean seasonal variability? Because these
Decadal
measurements, decadal, seasonal, it doesn't matter. In the poles, they are measuring the same, like, they don't measure there in the winter, you know, so they measure there in the summer. So
I'm talking about longer term.
40 years interannual variability, I didn't test it, I just, yeah, it's a good point, I should maybe try, but yeah, I'm happy to speak further. Can I?
First of all, very nice talk, very diverse. So you discuss variations of climate over a wide range of timescales. You talk about global warming, decadal or 40-year timescale versus geological timescales. But all of it centered around ecological processes. Could you for someone like me who doesn't really understand ocean chemistry or ecology, could you explain how come same processes affect the ocean on such short time scales?
It's a good question, yeah.
But also on such large time scales. I mean, evolution in some way should also get in the way. I mean, the basic properties of these animals should change also over time. Right?
Not only, also the processes that take place. For example, there are papers that show if you change the carbon to phosphorus ratio in the ocean today, you mute the CO2 in the atmosphere because you bury more oxygen to the deep ocean. Okay? So models predict that if you increase the C2P, more carbon will get into the deep ocean and less will be in the atmosphere. This is present day ocean. But when I did the Palair red fill ratio, I was also expected to see that higher red fill ratio, less PCO2 in the atmosphere relative to a fixed case. However, I saw that on geologic timescale, it's not, because there is other processes. Because you bury more carbon, but then you produce more oxygen. And then the oxygen on long timescale, it's not relevant to climate change, it's relevant to geological. And then more oxygen produce more oxidative weathering, so it reduces the carbon. So, for example, the atmospheric carbon dioxide is not sensitive to C2P over geologic timescales, but it is sensitive to CO2 on these timescales. So it's really interesting interplay. Yeah.
Thank you for a huge talk with lots of topics. I hope I got some of it. Could you explain why in a warmer world the 40 zone will deepen, which was
The biomass will deepen.
because temperature varies a little bit, but the drop in the depth of the 40 zone is appreciable.
Yes.
So,
This is a very good point. Okay, so you have interplay of processes. First, you get warmer. You have warmer surface. Phytoplankton growth is also sensitive to temperature. Mortality is sensitive to temperature. Higher temperature, you have more mortality. And then they go a bit deeper when temperature is more optimal for them. Also, you have enhanced stratification. So it's more hard for nutrients from the deep ocean to be delivered to the surface ocean. So biomass also declining to be a more optimal place between light and nutrients. So it's a combination of temperature and nutrients. Another point is also a thing of light. So because of phytoplankton in the surface are declining, the biomass is declining in the surface, a little bit more light penetrates, a little bit. And this gives the phytoplankton more benefits also a little bit.
So today, phytoplankton in the surface reduce the depth of the floating zone, they shape
They should go deeper. It has been observed in the Sargasso Sea, in the BAT station, that phytoplankton biomass shift deeper. And this is...
but the floating zone is dependent on the concentration of phytoplankton.
The photic zone, you mean the 1%? Yes, because it's your shading. They absorb light, so it changes the profile of the temperature of the light. And also of the temperature, because they absorb temperature. So this is a great future topic to explore, whether other models see this, and whether the observation, what implication for the depth of the photic zone.
I have a comment and 2 questions. Can you show me again the high latitude graph?
High latitude graph?
Yeah. Well, where you have the trends over
the
the
polar.
1970s.
Yeah.
Yeah, so I'm looking at the trends and I simply like.
You're not a biologist.
I'm not very convinced. But let's say it is true. So my question, that's my comment. So my question is, this is from the water body, right? So this has been collected, these are samples that have been collected from the water, right? Not from sediments.
No, from the water,
So-
surface water.
So my second question would be, what kind of, because this presumably is going to be partially transferred to some sediments that have at least some kind of organic matter in them, and what what would you expect to see in sediments if you were to take a core, going back to the question before about over kind of geological timescales, how can you test this on geological timescales, and what would your predictions be for what's going to be in sediment?
Okay, in polar region, I don't really know what is left in the core, but I know that most of the primary production is being recycled because there is not a lot.
Right.
So the marine food web is really efficient in recycling, so I don't know what will get preserved in the sediment. Yeah, so this, I don't know
So it's a
how
perfect crime.
Yeah, we cannot test it over geologic timescale. With this observation, we can indirectly test it, maybe in the amount of oxygen in the deep ocean, because lipids consume more oxygen. So to go to ocean anoxia, and then you can look in other geologic proxies for ocean anoxia. So indirectly, maybe you can measure it, but directly, I'm not sure.
Any more questions?
So it's a question maybe about something that you haven't done, but what you think might happen. So you kind of showed the Darwin model, or at least a version that looked to me like it was eddy resolving and looked really beautiful.
Yeah.
And then the simulations you're doing are like 2-degree resolution.
Yeah.
So do you have a sense of how like real ocean circulation with very complicated eddies and transport processes would affect biological processes that you're modeling here? Would currents, say, more realistically
It's a great
resolve
question.
without have large changes on anything?
It's a
Do you
field,
have a
but
sense of that?
it's a field by itself, and my group members are working on this. How like eddies impact nutrient input. Like you have like small scale eddies that change also the nutrient distribution in the ocean. And it's not my field, and I know that if we die higher resolution, I will have great movies. But other than this, I don't know how it will influence my model. And I don't think it will give us something more because there is not enough observation to validate it. So there is not really a reason to increase the resolution at this point. But it's a field by itself how the fine scale eddies impact biological productivity. Yeah.
Any more questions? Okay, so let's thank our speaker.
By the way,